So, hello and good morning or good afternoon, depending on where you are. My name is Matthew Cobham, and I'm responsible for lighting application, indoor lighting application at Philips Lighting. And on behalf of the Philips Lighting University, and in collaboration with the Parsons New School for Design, I would like to thank you very much for attending this webinar called Light and Time, the Discovery of a Third Photoreceptor System Within the Eye. Now, this is actually the second of our three-part series under the subject of light and well-being, and our presenter today is Professor Russell Foster. So let me introduce Russell to you a little bit for those of you who don't already know his work. Russell Foster is Professor of Circadian Neuroscience and Director of Sleep and Circadian Neuroscience of the Circadian Neuroscience Institute at Oxford University. In 2008, Russell was elected as a Fellow of the Royal Society, and in 2013, he was elected as a Fellow of the Academy of Medical Sciences. He has received several distinguished scientific prizes and given numerous distinguished lectures around the world, and is chair of the Cheltenham Science Festival, member of the Council for Biotechnology and Biological Science Research Council, the BBSRC, and member of the Governing Council of the Sainsbury Wellcome Centre. In March 2012, he was named Social Innovator of the Year by the BBSRC. Russell's broad research interests consider the mechanisms that regulate and generate circadian rhythms and sleep, and what happens when these systems are disrupted by the environment and brain disease. He has published nearly 200 scientific papers and has co-authored 3 popular science books: Rhythms of Life, Seasons of Life, and most recently, Sleep, a very short introduction, published by Oxford University Press, and he is currently working on his 4th book. Now, before I hand over to Professor Russell Foster, I'd like to just remind all of those of you who have logged in, and actually there's really quite a large number, well over 200 people, so that's really great to see. Just a few sort of small technical things. On the right-hand side, there's kind of a question and answer box, and please type in any questions that you have for Russell Foster in there, and we will do our best to answer at least some of those at the end of the webinar. Also, you can also direct any kind of more technical questions regarding the running of your system to the host, if you like, of the webinar. So, without further ado, I'd like to give a very warm welcome to Russell Foster. It's great that he's joined us today to give this webinar, and I'm looking forward to it very much myself. So, let me hand over to Russell Foster.
Many thanks indeed, Matthew, and also for Philips for sponsoring this series of 3 lectures. Well, welcome everybody to this session on Light and Time, the discovery of the third photoreceptor system within the eye. Before I kick off, I would like to acknowledge, I'm just trying to change the slides here. Advice team, I can't advance the slides. Welcome again, everybody. We've just worked out how to advance the slides. So, welcome again to Light and Time, the discovery of the third photoreceptor system within the eye. Let me, first of all, acknowledge the people who funded much of the work I'm going to talk about, the Wellcome Trust, the BBSRC, and the Oxford Biomedical Science Research Centre. And I'm also going to acknowledge right at the beginning some key individuals, Mark Hankins and Stuart Pearson, with whom I've collaborated to produce much of the work you'll hear about. The human work will be by Katharina Wolf and Susan Downs. Steve Hughes and Artie have been critical in some of the cellular work we're doing, but I won't show too much of that today. And of course, in the past, Iggy Provencio, Bobby Soni, and Rob Lucas. I've structured the next 40 minutes or so into the following sections. First of all, mammals and the photoreceptors of the eye, and we'll kick off with a discussion of the image-forming photoreceptors, classical vision. And then what I'd like to do is compare it with non-image detection and, in fact, the discovery of this third photoreceptor system, and how important it is in regulating things like the biological clock. Then we'll pop in and discuss some of the spectral responses that you see with these new photoreceptors and introduce melanopsin, this new photopigment, and then finish this first section by discussing different photoreceptor interactions. Now, much of the work in the first section has been undertaken on mouse models, and so I think it's then important to try and extrapolate and ask the question, to what extent can we extend the work and the findings from mouse into the studies in humans? So we'll talk about that, we'll talk about spectral responses, and we'll also touch on irradiance levels, how bright the environment has to be to stimulate some of these photoreceptors. And then finally consider some of the clinical implications of these new receptors and the impact it's having on, indeed, clinical ophthalmology. Okie dokie, let's go to the first section, and image-forming vision. Well, of course, this extraordinary structure, the human eye, performs two very different light-detecting tasks. The familiar task is, of course, image detection, the ability to identify an object against its background. And we can do this either in black and white, or, of course, we can use color to identify an object against its background. Now, that photosensory task is dramatically different, as I hope to convince you, of the second light detecting function of the eye, which is brightness detection. Essentially, we're not looking for an image here, we're looking for a gradient in a photon flux, a gradient in light intensity, and indeed color is frequently irrelevant in that light detecting task. Let's start, though, with a reasonably in-depth discussion of what we mean by image detection. And we'll do this with reference to the mammalian eye and, in fact, the human eye. So here we see the classic image. Here's the eye, and we've taken a slice out of the eye here, and we see the classic image of the retina. And here's the pigmented epithelium. Here's the photoreceptor cells of the eye, the light-detecting cells of the eye. Here's the inner retina, which is the part of the retina which undertakes the first processing of the visual image before a decision is made to actually trigger an action potential in these ganglion cells and then fire this message down the optic nerve and into the brain. Now, the light-sensitive photopigments that we'll talk about in a moment are housed, of course, in the outer segments of these rod and cone photoreceptors. So let's zoom in on this in a little bit more detail. Here we see one of our rod photoreceptors, and we've expanded it up here. And what you see in this outer segment are a series of membrane stacks. And embedded in these membranes is, in fact, the light-sensitive molecule, which is, of course, called a photopigment. Now, in the vertebrates, the light-sensitive molecules all have the same basic biochemistry. And so you have this 7-transmembrane protein called an opsin. It sits in the middle of the membrane, forming a sort of cage in which a very specific form of vitamin A, 11-cis retinal, is embedded. And the first stage of light detection is the absorption of photon by 11-cis retinal. It undergoes a conformation change and stretches out. That changes the disposition of these alpha helices, and ultimately that triggers a cascade that leads to a change in membrane potential in this photoreceptor cell here. Now, there are lots of different opsins producing a great variety of different spectral sensitivities, a great variety of different photopigments. So we can have photopigments peaking in the blue part of the spectrum, and we'll see some, in fact, ultraviolet pigments later on, in the green, in the yellow, and of course the red. And one critical feature I want to point out to you is that although the wavelength of maximum sensitivity of these different pigments is different, the shape of this absorption spectrum is the same in all of these different pigments. So if you have a response which could be described by a curve like this, you know immediately you're dealing with a photopigment molecule like this, based upon an opsin and a vitamin A. How an opsin produces this spectrum of photopigments, or how evolution generates these different pigments, is that you can change the amino acids that line this pocket here. Those amino acids carry a different charge, and they will have an effect upon the stability of the 11-cis retinal. And essentially, it's the charge pocket which dictates where the 11-cis will absorb photons, in the blue, the green, or the red. Now, human visual pigments come in 4 different types. There's a shortwave pigment peaking at 420 nanometers. There's our rods peaking at 500. Then we have 2 cones which are very closely related. One at 530 nanometers, and in fact a second cone peaking at 560. It's important to stress that, red is a very important color to us as a species, but we have no dedicated red pigment. But I'll explain in a moment how we see red light. Okay, we use our rods essentially to generate a black and white image of the world for simple contrast detection, and of course we tend to use these rods and under low levels of light. And although the pigment peaks at 500 nanometers, we have no knowledge that it's a green sensitive pigment. It's essentially the ability to detect light or not. To see color, you have to compare the output of different types of pigment. And that's what we use our cones for. Now, I hope all of you here can see this diagram. And what you should be able to see is the letter 8. Now, if you can't see the letter 8, then either your computer screen is not working properly, or you have some form of color vision. How do we see this 8 in this background green here? Well, we have a pigment at 420 and 2 pigments at 530 and at 560. How do we use those? Well, how do we see this blue line here? Our short wavelength pigment would absorb, and it will be stimulated around about 32%. The next pigment would absorb at a much higher level, at around about 80%. And our longest wavelength pigment would stimulate at around about 40%. And so what the retina and the brain does is take the relative outputs of these 3 different spectral channels, and that 80-40-32 encodes this particular color blue. And just to make that absolutely clear, if we were wanting to detect this particular color, then our short wavelength pigment has no real detection way out here in the orange part of the spectrum. Our first green pigment at 530 really will only have a minimum detection, about 15%. And our slightly longer wavelength green pigment will have a detection level of 40%. And so an output of these 3 receptors of 40, 15, and 0 encodes the color orange. Okay, I hope that's given you some sense of how our image-forming vision works. And what I'd like to do now is contrast that with non-image forming photoreception. Okay, so essentially gradient light detection. So let's now talk about brightness detection. Well, of course, it's very clear why we have image detection. Non-image forming detection, such as the detection of overall levels of light within the environment at twilight, can be used for a whole variety of different tasks. There's the regulation of the body clock, and we'll discuss that in some detail in a moment. The regulation of seasonal biology. Our levels of alertness and mood can be regulated by light levels. Endocrine cells can be directly regulated by light, and indeed even part of our pupil constriction, as we'll see, will be measured by, will be regulated by the overall levels of brightness detected within the environment. Let's now consider the big topic, which is the regulation of the body clock, and how this led to the discovery of a third photoreceptor system within the eye. Well, a little bit of background about your body clock. And of course, many of you will be more than aware that if we look at 24-hour changes in human biology, in the middle of the day, in the middle of the night, every parameter we care to assess, whether it's alertness, melatonin, this hormone that I'll talk about in a little bit from the pineal, growth hormone, body temperature, digestion, muscle strength, all show a dynamic 24-hour physiology. Let's just look at two of these parameters in a little bit more detail. If we look at our levels of alertness, which are reasonably stable over the day, but towards the end of the day and the beginning of the night, they start to drop, such that at 4 o'clock in the morning, our ability to process information is as impaired as if we'd consumed sufficient alcohol to make us legally drunk. So our ability to perform at that time of the day, we might intuitively know is not great, but actually is severely impaired. Now, that's not due to solely to fatigue. Fatigue will make it worse, but essentially it's this gating of our brain by this internal clock. If you look at also core body temperature, we see about a degree variation. Again, the low point at around about 4 o'clock in the morning. And if you're an athlete, this can have some significance. So, for example, if you look at the athletic performance of a swimmer, during the sort of early hours of the morning, let's say 5 o'clock in the morning, 6 o'clock in the morning during the training session, versus later in the day, 8 o'clock in the evening during the competition, they can swim 100 meters in the pool 2.7 seconds faster here than they can here. Now, the point I want to stress is, of course, if you exercise, you'll change core body temperature, but it will be superimposed upon this moving endogenous baseline. And that's the key point I'd like to make, is that all of this physiology we've been talking about will persist, or this rhythmic physiology will persist under constant conditions of temperature and constant darkness. Now, the origins of this day within, of course, are in the brain. And here we have the eyes with the optic nerves forming the optic chiasm. Here's a little pituitary gland. And just above where the optic nerves fuse, the optic chiasm, are these two structures here called the suprachiasmatic nuclei, mercifully abbreviated to the SCN. And each individual SCN cell, and each individual SCN cell is indicated as a yellow spot here, is capable of generating its own 24-hour oscillation. And what I mean by that is you can take this cell out, put it in a dish, record, put an electrode in it, and you can record 24-hour oscillations of electrical activity from that single neuron, telling us that the clock is not the property of cell-cell interaction, but of a molecular mechanism. And we don't have time, unfortunately, to talk about the molecular basis of the clock, but it's one of the great achievements, I think, of circadian biology. You have about 50,000 of these individual clock cells all working together to produce what has been called the master body clock. And let me stress, if this region of the brain is damaged as a result of a tumor, for example, and we studied a gentleman who had a tumor which advanced through this region of the brain, and he lost all of his 24-hour circadian rhythms. And so this is the key master clock within the brain. Okay, what we used to thought happened is that the master clock would produce a signal that would impinge upon the body and drive 24-hour oscillations, so the sorts of things that we've been talking about, rhythmic changes in core body temperature and alertness and all the rest of it. But in fact, our understanding has changed really quite markedly over the past few years, with the appreciation that most cells in the body have the ability to produce a 24-hour oscillation. So there are literally billions and billions of individual oscillators clustered into the various organs of the body. They receive a signal from the master clock, and we often don't know what that signal is, which then coordinates this entire circadian or this body clock network. Now, the whole point of this master clock and all of this circadian biology is to fine-tune physiology and behavior to the varying demands of activity and rest. And the way that you lock on internal time to the external world, of course, is with the eye, which is detecting the light-dark cycle. Now, many of the rhythmic processes that are depicted here are often illustrated in the form of what's called an actogram. And we see here a cartoon of an actogram. So essentially, each line represents a day, and in this particular cartoon, wake is the thickened portion of the line. the point of this illustration is that with the light-dark being detected by the eye, which then sends a message to the SCN, which then aligns internal physiology to the light-dark cycle. And the key point to make is that if you have no eyes, this ability to regulate internal time is lost completely. You don't destroy the clock, of course. The clock will keep on ticking, but in the case of the human body clock, it's slightly longer than 24 hours, about 24 and a half hours. So we'll get up later and later and later and later each day, because we are simply, without our eyes, unable to align this internal time to the light-dark cycle. Now, some of you may have become aware of some old work suggesting that light behind the knee is capable of entraining the body clock. That turned out subsequently to be nonsense. And the latest incarnation that there's a non-eye photoreceptor in humans as a result of light being squirted into the ear and illuminating the brain. And the argument there is that can also regulate the clock. I have to say the jury is very much out on that, and in fact there's very little solid evidence at all that we have anything other than a photoreceptor within the eye. And in fact we're exploring this whole idea that light in the ear can stimulate the brain with some individuals who have no eyes. And watch this space. I'll give you an update on that as time progresses. Okay. Now, one of the key things I want to stress is that although both circadian light detection, the alignment, the entrainment of the body clock, and visual responses are mediated by the eye, the way in which the eye processes this light information is very, very different. The visual ecology of these two photosensory tasks is markedly different. So for the circadian system, what it needs to do is sum light information of the entire environment. It's detecting global irradiance. Well, of course, for vision to work, it must capture light information from a very specific region in space, classically a task called radiance. The circadian system is broadly insensitive to dim light, whereas, of course, the visual system is ultra-sensitive to dim light. In fact, light at which we can comfortably read by will not be effectively detected by the circadian system and will not affect the clock. The circadian system is insensitive to relative short-duration stimuli, whereas, of course, the visual system to work is only sensitive to short-duration stimuli. And finally, of course, the circadian system integrates light information over very many minutes, whereas the visual system integrates light information over the millisecond range. So, for example, you can give the same number of photons squeezed into 5 minutes or expanded out over 45 minutes, and the clock broadly reads this as the same signal. These very different sensory tasks puzzled me. I couldn't understand when I first started to think about these questions how the classical visual system could also reliably get overall measures of the amount of light in the environment and extract, for example, time of day information. It just didn't seem to make sense how the classical visual system could do that. And so we started to look at the mouse eye, and the mouse eye is very much like the eye I showed you earlier. Here are the rod and cone photoreceptors. Here's the inner retina, ganglion cells going off to the optic nerve. And what we decided to do was look at, in fact, 3 different mutant mice, and they're illustrated here. The details are unimportant, but the net result of these mutations, or in fact this one is a transgenic animal made by Maureen McCall, is that the rods and the cones had largely degenerated, leaving pretty small numbers of pretty rudimentary photoreceptors left. They lack an organized outer segment, and they were very, very poorly preserved. The key point is these mice had no image detection left. They had their eyes, of course, but as a result of the rod and cone loss, they had lost their ability to form a visual image. We then placed these mice into a little running wheel here, connected it up to a computer, and here's one of those actograms again. And again, we'll see examples of this as we go through the talk. So just for clarity, here's a light-dark cycle. Of course, the mouse is a nocturnal animal, so each day here it's running during the night, and we see it's beautifully entrained. It's entrained to the light-dark cycle. We can then plunge the mouse into complete darkness. Now, the mouse body clock is slightly shorter than 24 hours. So instead of us getting up later, it will get up early and early each day. The key point is that you can turn on the light in the middle of the dark very shortly after the animal started its activity, and the next day it'll get up later. It produces what's called a beautiful phase shift in the free-running rhythm of activity. Okay, so let's look at the data. This is the assay. This is a perfectly normal wild-type retina. Here's increasing the brightness of our little 15-minute pulse of light here, and this is the size in minutes of the shift we saw over the following few days. Normal retina, normal dose response curve, that's what you'd expect. This is a second sort of mouse with one copy of the defective gene, but broadly speaking, the retina is normal. It's not a degenerate retina. Here's our degenerate retina, two copies of the defective gene, and we see a nice irradiance response curve. And what was truly remarkable in these early studies is that when you overlay these three irradiance response curves, they are statistically indistinguishable. And this was the first bit of data which suggested we may be onto something really very exciting here, because what we've got is normal circadian responses to light in a mouse which was functionally blind, had no image detection at all. Now we know this receptor was located in the eye because if you use opaque contact lenses blocking all light reaching the eye, then the ability of the eye to detect this light is gone, and the animal will just drift through the rest of its life free running. Okay, so with these early data, we wondered that maybe there's something else. Could there be another photoreceptor within the eye? After all, you can regulate the clock perfectly well without these classical photoreceptors. When we proposed this, the opposition from vision neuroscientists was particularly ferocious, and the opposition is summarized in the following slide. It was hard to imagine, somewhat 15, 20 years almost later, the ferocity with which some of this data was initially received. I remember in one seminar, I showed data like I've just shown you, and somebody at the back stood up, said bullshit, and just walked out. Now, the criticisms, where they were constructive, were broadly of two forms. The first was that we've been studying the eye for over 150 years. Are you seriously telling us that we've missed an entire class of photoreceptor within the eye? So you're talking nonsense, Foster. Well, we could address that, not in mammals to begin with, but we were doing parallel work in fish. And Bobby Soni, my PhD student, identified a very interesting gene which we called vertebrate ancient opsin. It was another one of those opsin photopigments. But what was so extraordinary about it, excuse me, is that it wasn't located in the rods and cones, but it was located within a small number of horizontal cells and ganglion cells. And it would produce a light-sensitive pigment somewhere between 460 and 500 nanometers. So what we had fairly early on, not from mammals, but from fish, was proof of principle. It's not such a stupid idea to suggest that the eye of the retina might contain another class of photoreceptor in mammals, because we'd already shown that in another group of vertebrates, the fish. Another and semi-reasonable criticism of our early work was, well, actually, your models just weren't good enough, because they didn't entirely eliminate all the photoreceptors. You still had those rudimentary photoreceptors left. And so what was happening is that the circadian system could probably maintain normal photosensitivity with reduced numbers of rods and or cones. And so clearly we had to do better experiments, and what we did was take our naturally occurring mutant animal in which the rods had degenerated and combine it with a transgenic animal in which all the cones had been eliminated. You eliminate all of the rods and cones. And in the next couple of papers, again, same assays that I've showed you before, the ability to shift the clock, and to our intense pleasure, these are the increasing brightness along here, the size of the phase shift here for a mouse with absolutely no rods and cones, and here's its wild-type compatriate, no statistical difference, or there are subtle differences which we'll come back to later, but no statistical difference whatsoever. Another assay we used in these early experiments was also to look at the regulation of pineal melatonin. This is the hormone that is released from the pineal gland, and the pineal is regulated by the body clock, the SCN. And what happens is that there's a rhythm in melatonin secretion, as indicated here, and the rhythm is always at night, and that's regulated by the body clock. But you can also turn the lights on in the middle of the night and acutely suppress the release of melatonin from the pineal. And what we were able to show is that in our mice with no rods and cones, you could fully suppress pineal melatonin in the absence of any rods and cones at all. So with these two back-to-back science papers, we were able to say that the RD mouse could allow us to conclude that the third ocular photoreceptor must exist within the mammalian eye, and this model provided us with a conceptual framework to address what these photoreceptors might be, and indeed what else they might do. Okay, so if it's not the rods and cones, what other choices do we have? Well, it could be horizontal cells or ganglion cells, because they were the two classes of inner retinal neurons that we were finding as light sensitive in fish. But in mammals, it's not the horizontal cells, it's the ganglion cells. About 1 in 100 of the ganglion cells, and these are the cells that fire off their projections and form the optic nerve, about 1 in 100 of those are directly light sensitive. And we call them photosensitive retinal ganglion cells. This is our third class of ocular photoreceptor, the rods, the cones, and the PRGCs. This was shown by using classical techniques in the rat by David Burson, at the same time by us using the rodless coneless mouse, and I'll explain what we did in a few moments. And a few years later, some beautiful work by Dennis Dacey on the macaque. And essentially all three studies showed that there are photosensitive ganglion cells in these groups of mammals. As I say, our approach was to use the rodless coneless mouse, and we combined that with calcium imaging. So you can take this retina, put it in a dish, load it up with a calcium-sensitive dye, and then turn the lights on in isolation. And you see that individual neurons will turn on in response to light. Now, again, I don't have time to talk about the physiology of these cells, but I do want to mention that they are not simple cells. What we found right from the beginning, way back in 2003, is that here's the light going on for about a minute, and this group of cells produces a sustained response. They're essentially responding to light while the light is on. The second group, a different sort of ganglion cell, we think, was a transient responder. It was actually turning off while the lights were still on. And a third remarkable group, where the lights were on for about a minute, and they still would signal light exposure for 20 minutes afterwards. So don't run away with the impression that this is a very simple system. And in fact, much of our recent work has been looking at the cell biology and the molecular biology of these cells, and they are remarkably complicated and deeply fascinating. But that's, again, perhaps for another story. The other thing that emerged fairly quickly is that these photosensitive ganglion cells don't just project to the master clock, the SCN, but they project to multiple structures within the thalamus and the hypothalamus. This was work that we showed originally with viral tract tracing with Howard Cooper, but much more elegantly and more recently with Sama Hatter's beautiful illustration of the projection of these cells to various brain structures. In terms of function, what are these projections doing in these different structures? Well, of course, we've already talked about the projection to the SCN, regulating rest activity cycles and the melatonin profile. But there's a projection to the ventrolateral preoptic nuclei, the VELPO, and that's critically important in the regulation of sleep. In fact, another projection to the dorsal geniculate nuclei seems to be very important in the suppression of activity. This is a mouse. The lights have been turned on, and you see that it's stopped running. And that acute suppression of activity in response to light seems to be mediated by these remarkable cells. And even part of our pupil constriction. There's a wonderful projection to the livery pretectal nuclei, and we showed this first of all in mice and then in humans, and that these cells are regulating part of our pupil constriction. In fact, let's just illustrate the pupil constriction in the mouse in a little bit more detail, because we'll talk about this in a moment in some other assays. So what we see here is a fully dilated to the fully constricted pupil of the mouse. It's sitting in the dark, and we're filming this using infrared vision goggles. The lights have gone on, and it's constricted. So increasing the brightness of the environmental light, going from fully dilated to fully constricted. What was remarkable was that if you do the same experiments in a mouse with no rods and cones, it needs more light, but it can fully constrict the pupil in the absence of the rods and cones. So we're not dealing with just a circadian photoreceptor here, but a generalized irradiance detector mediating tasks as diverse as pupil constriction to regulating the body clock. Let's now drill down and ask a bit more about the photopigments, the spectral responses of these photosensitive ganglion cells. What's the light-sensitive molecule of these PRGCs? Now, the known mouse photopigments at the time, different from humans, of course. Mice have an ultraviolet sensitive pigment peaking at 360 nanometers. We don't have any ultraviolet sensitivity in that range. They have a single rod, of course, just like us, and they then have a second cone. So, unlike us, they've only got two classes of cones. We have three. And so the question was, could these photopigments, could these opsins be located in the photosensitive ganglion cells and be regulating the sorts of light detecting tasks that we've been talking about? And so what we did was an action spectrum. Essentially expose mice to different wavelengths of light, and these were mice with no rods and cones, the RDCL mouse, and then look at the effect of different wavelengths of light and their ability to cause pupil constriction. And to cut a long story short, we defined a beautiful opsin vitamin A pigment. We knew it was opsin vitamin A because that's the standard absorption for an opsin vitamin A, and it fits the data absolutely beautifully. But the lambda max, the wavelength of maximum sensitivity, is at 480 nanometers. It's not where the known pigments of the mouse eye were peaking, the ultraviolet, the rod at 498, and the long wavelength at 508. That was the pupil. What about shifting the clock? Again, same sort of protocol. Change the wavelength and intensity of this 15-minute pulse. Look at the size of the shift. You generate endless irradiance response curves. The bottom line is the peak sensitivity is at 480 nanometers, completely different from the known classes of the rods and cones. So it looked as though there was another spectral channel. It was another type of opsin-based photopigment, peaking at 480 nanometers. Now, when we got these data, of course, our first thought was, well, maybe it's VA opsin. That was that photopigment I talked about in the fish eye, turning up in horizontal cells and ganglion cells. But actually, VA opsin is not expressed in the mammals at all. It's expressed in lots of other vertebrates, but not the mammals. In fact, of course, we all now know that the new photopigment, peaking at 480 nanometers, is melanopsin. Now, melanopsin was discovered by Iggy Provencio. Actually, Iggy was my PhD student back in the days of Virginia, and Iggy then went off to work with Mark Rolag at Uniform Services, a remarkable chap, and when he left the lab, he said, I'm never going to work on mice again. They're smelly, they bite me, and they're expensive, so I'm going to work on a really lovely creature like the African clawed toad. And what he was interested in is how the pigment cells in the skin of this toad will contract in response to direct light illumination. And Iggy found an opsin in these melanophores, these light-sensitive pigments, and he called it melanopsin. And of course, that kicked off a wonderful line of study, because of melanopsin was then found to be turning up in these light-sensitive ganglion cells, and this is one of those light-sensitive ganglion cells stained with an antibody against melanopsin. And you can actually record beautifully light responses from these melanopsin-expressing ganglion cells. Okay, so we knew it had to be an opsin. We got an opsin in a light-sensitive ganglion cell. How do we definitively link melanopsin to the capacity of these cells to detect light? Oh, and this just shows a wonderful picture of multiple photosensitive ganglion cells expressing melanopsin. And they form sort of a net, a light-sensitive net across the arc of the eye. Okay, so how do we know it's melanopsin? Well, lots of studies have been undertaken. I just want to mention the functional ablation studies. And these were a great collaboration between my group in Qinghua Yao and led by Sam Hatta and Rob Lucas. And the experiments are summarized as follows. If you remove the rods and cones and you take away melanopsin photopigment, the cells are still there, but they lack any melanopsin. And you look at, you abolish all light responses. the mice with no rods, cones, or melanopsin, and I should say melanopsin is often abbreviated to this symbol here, OPN4, no ability to constrict the pupil, and no ability to entrain the body clock to the light-dark cycle, or indeed shift the body clock in response to intense pulses of bright light. So the rods, the cones, and melanopsin can account for all of the light-sensitive function of the mammalian eye. But what we thought would happen is that when we took away melanopsin, we would abolish all light responses. Remember, those early studies suggested that the rods and cones were playing no role whatsoever, but we were wrong. What you see is greatly attenuated irradiance responses, but you still see responses. So here's a mouse with no melanopsin. You can't fully constrict the pupil. You can only go to that state of pupil constriction. And that tells us, of course, that melanopsin is required for that last bit of pupil constriction under bright white light. And the bigger surprise, I think, was the fact that you can still entrain the clock to a light-dark cycle and still get small, but highly significant phase-shifting responses to a pulse of light. So, there's a complex and important interaction between rods, cones, and the photosensitive ganglion cells. In the absence of melanopsin, the rods and cones can partially compensate for the loss of melanopsin. And in fact, if you think back, we originally conceived these two systems, the irradiance detector and the image-forming receptors, as parallel, perhaps not talking to each other at all. And now we know that's not the case. The two systems do talk to each other. And in fact, that's the topic I'd like to consider now, these photoreceptor interactions. And I should say, this is turning out to be one of the really hot topics, I suppose, in the field at the moment. Let's just recap. We have three classes of rod and cone cell: rods, a green cone, and an ultraviolet cone in the mouse eye, and our melanopsin OPN4 cells. Oops, yes. We also know, that the rods and cones can talk to the melanopsin ganglion cells via bipolar cells, and indeed via horizontal cells. There's a little chit-chat between the classic photoreceptors and these photosensitive retinal ganglion cells. So we know they talk to each other. And a series of papers have come out describing how they might talk to each other. And I'd like to show you some of the work that we've been doing with Jokke Meyer from Leiden in the Netherlands. This has been a terrific collaboration. Basically, what you can do, and what Jokke has perfected, is the ability to place a recording electrode into the SCN and record the electrical activity of a freely moving animal. It's wandering around in its cage. We can illuminate the eyes, and they may have normal eyes, or they may have lost their melanopsin. So we have lots of flexibility on the type of stimulus we can send off to the SCN. In a normal wild-type animal, you see the resting potential here. You see the lights going on, an overshoot, a sustained response, the lights go off, there's an undershoot, and then back to baseline. And we'll see examples of this in the next few slides. So what you do, what you do when you've recorded from the SCN is get this here's a cartoon version of what I've just shown you: on response, sustained, off response. And what had sat in the literature until very recently was the sort of following argument: that it's the rods and the green cones that are producing these transient responses here. But it's the sustained response is being produced by melanopsin ganglion cells, such that if you take away melanopsin, you still get the transient responses, but essentially you've lost the encoding of irradiance information at the level of the SCN. Okay, but nobody had looked at the role of the ultraviolet cones. So what are these ultraviolet cones doing, and how might they talk to these photosensitive ganglion cells? And this is a paper we published just towards the end of last year, and I'd like to acknowledge four remarkable young individuals, Flor, Simon, Hester, and Tom, who all contributed a terrific amount of work to this publication. So our fundamental question is, what's going on? What's happening with these ultraviolet cones, and how might they regulate the SCN? Our prediction was as follows, that with a normal retina, you'd illuminate the mouse with light, and the only cells capable of responding to ultraviolet light would be ultraviolet cones, and so you get a nice transient on. That's what we predicted. But the melanopsin ganglion cells aren't sensitive to ultraviolet light, and so they would show very little sustained response. So it would be rather like knocking out melanopsin. Stimulating with ultraviolet light would produce the transients, but no sustained response. What we actually found was the following result. UV stimulation in a wild-type retina. We got transients and sustained, and transients offs. So it looked as though the ultraviolet cones could produce both the on and off responses and the sustained response, which had been attributed exclusively to melanopsin. So is it the ultraviolet cones that are mediating these on, sustained or off, or could it be that melanopsin is also capable of detecting ultraviolet light? And of course, the way we solved that problem was to do the experiments in a mouse with no melanopsin, and there was no change. So melanopsin is not sensitive to ultraviolet light. This response is being driven by the ultraviolet cones. So it's all very complicated. These photosensitive ganglion cells, based upon melanopsin, and the ultraviolet cones are playing a very similar role in irradiance regulation of the clock. They're producing transient on, sustained, and off. How do they do this? We've absolutely no idea at this stage. How can messages from the ultraviolet cones talk to the photosensitive ganglion cells and essentially mimic the sustained responses you get from melanopsin? Which raises the question, what's special about melanopsin and the photosensitive ganglion cells? If you can encode irradiance information with an ultraviolet cone, why on earth would you need a photosensitive ganglion cell based upon melanopsin? And these are questions we don't have answers for at the moment. Okay, let's now move on and ask the question, to what extent is the information that we've learned from mice relevant to studies in humans? Do humans lacking rods and cones also retain these non-image forming responses to light? And this is a paper I very much like, because it's one of those rare examples where there's some sanity in science. I turned up to a meeting presenting our work on a lady with no rods and cones, and Steve Lockley turned up to the same meeting reporting their data from a gentleman who had no rods and cones. And of course, we didn't run back to our labs and try and publish first. We fused the two data sets together. And I've highlighted here in blue the people from the UK side of things, and in black are the Harvard side of things. And Farhan Zahidi from the UK led this study. So our patient, SC, was an 88-year-old lady with an undiagnosed gene defect in her eye, which had resulted in the loss, as far as we could tell, of all of her rods and cones, with no perception of light for well over 50 years. Her fundus looked like this. It was a complete and utter mess. And optical coherence tomography showed that we could find no evidence for any residual rods and cones. When Farhan presented this genetic case, he's fundamentally interested in eye disease, I asked him, well, does this lady regulate her body clock to the light-dark cycle? And his response is, well, I'm an ophthalmologist. Why would I ask such a stupid question? But he went back and he called her up and said, well, SC, do you get up and go to bed at about the same time every day? And she said, yes, no problems at all. So Katharina Wolf then studied SC and showed, and here we have another actogram. This is from a human, slightly different in this case. Here's midnight to 6 o'clock in the morning. Here's the dark band, grey band here. And day one, day 2, day 2. We've double plotted it, so you can see the pattern over 48 rather than just a 24-hour period. The bottom line is that she could regulate, for an 88-year-old, her rest activity profile remarkably well. She's not showing a drifting pattern that you would expect if she couldn't endrain. There's some activity in the middle of the night, but that's very typical for an 88-year-old individual. The Harvard man with no rods and cones was younger and so had a much more robust rest activity cycle, but basically the same as our elderly lady. No evidence of a drifting clock, beautifully entrained, and yet no rods and cones in this individual. And of course, Steve went on and showed that not only was the rest activity entrained, but also the peak in nocturnal melatonin was also beautifully aligned. No evidence of a free-running clock. Okay, so looks as though the work that we had done in mice is supported with humans. We then went on and explored SC in a little bit more detail. What was really interesting is that she also showed a pupil constriction. When they first looked at her pupil, they thought she had no pupil constriction at all, and we then pointed out to the ophthalmologist, well, remember, the response of these photosensitive ganglion cells is relatively slow and sluggish. Leave the lights on. And don't just flash the torch in front of her eye. Leave it there for some time. And so she showed a slow but sustained pupil constriction. Very excitingly, when we did an action spectrum from that response, it peaked at 481 nanometers. Now, of course, it's 481 where melanopsin in the mouse was peaking, and of course it did not correspond to the known peaks of the visual system, the rods and the cones, suggesting very strongly that this was being mediated by a melanopsin-based photosensitive ganglion cell system. So, indeed, humans are like mice. That was all very pleasing. We also went on and asked her another set of questions. We asked SC if she was aware of bright light at some level, and she said absolutely not. I have no conscious light detection at all. But we asked her to perform the following task, which is the two alternative forced choice procedure. Essentially, there's an auditory cue every 5 seconds, and in a 10-second episode, the light is either on or off. And we simply asked the individual, was the light on the first or the second episode? We asked SC to perform these tasks, and she said, well, there's no point. I simply won't be able to detect the light. And we persuaded her and said, look, just imagine if you could see the light, can you think of when it might be on and when it was off? And when she performed the experiments, she got it 20 out of 20 times. No problem whatsoever. So she had the ability to perceive light, even though she was unaware of that light perception. Now, of course, the problem we faced here is, well, is this some residual rods and cones? We were back almost 15 years where we started with our mouse studies. And so the way around that problem was, of course, to do an action spectrum, to explore the effect of different wavelengths of light in this 5 seconds of lights on. So we compared different wavelengths, as you see down here, at threshold intensities of light. And what was very exciting is that it was only at a wavelength of 481 nanometers, at threshold intensities, could she perceive that the lights were on reliably above chance, supporting, in fact, that we have a melanopsin system in this eye that was perfectly well working, and also mediating yet another irradiance detection task, our awareness of light, but an unconscious awareness of light. Now, there's some confusion in the literature that melanopsin has a peak at 460, and I thought I'd take this opportunity just to clarify that. The early studies from Budd and from suggested a pigment around about 460 nanometers, and from Deborah's group, again, suggested about 460 nanometers. There are different ways of plotting the data, and the ideal way of plotting it is on not a percent sensitivity, but on a log transformed axis. And when Bud and Deborah very kindly gave them our data, gave us the data, and when we plotted it on a log sensitivity basis, the best fit was for a pigment at 480 nanometers. So there's no contradiction. The data that we've got from the melatonin suppression work from Bud and Deborah also peak at 480 nanometers. So there's remarkable consistency between both our results in humans, other people's results in humans, and indeed mice. So let's also just briefly touch upon irradiance levels. Environmental light, of course, has a huge dynamic range, from 100,000 lux on a bright sunny day to 0.25 lux in cloudy moonlight. But outside, even on a cloudy day, you're still getting to 10,000 lux, very much more than typical office lighting setting conditions. And the big question has been, how sensitive is the human circadian system to light? And the suppression of pineal melatonin in humans has been used as a way of trying to assess that. So there's a light pulse in the middle of the night, and the acute suppression of melatonin is used as an assay, and how much light is needed to drive this acute suppression down. As I say, it's a surrogate measure of sensitivity of the human circadian system. Now, the classic work in this area was done by Al Louis way back in 1980, and he said in his early studies that you needed around about 2,500 lux to fully suppress nocturnal levels of melatonin when you turn the lights on for 2 hours. Other studies are rather different. More recent studies by Christian Hjochen and Jamie Zeitzer have used 6.5 hours and suggested that half maximum responses are around about 100 lux, and that was consistent between Christian and Jamie Zeitzer's data, but full saturation at around about 1,000 lux. I'll just show you those data. This is for the suppression of melatonin, about 100 lux for a half maximal response, and about 400 lux, about 400 lux for a saturating response. A little bit different for shifting the rhythm in melatonin. So this is looking at the rhythm of melatonin and seeing how you can shift it with different intensities of a light-dark cycle. Half maximum response is 200 lux, so you need more light to shift the rhythm and significantly more light to saturate that shifting, that light effects on the clock. Bottom line is that for humans, we're talking about 1,000 lux for several hours to saturate circadian responses to light. So the statement that we started with, that the circadian system is broadly sensitive to light, is upheld. So it's significantly greater amounts of light than that one experiences in normal office lighting conditions, but of course much less than you get even on a cloudy day. And again, that's something we may want to discuss during the discussion period. Let's now finish with a few comments about the significance of these findings to ophthalmology. The key point, of course, is that visual blindness need not result in the loss of all light detection by the eye. Sleep and 24-hour rhythms, however, and abnormalities, however, are completely ignored in clinical ophthalmology. And let me just illustrate some of the work that we're now doing in the Oxford Eye Hospital, led by Susan Downs, who's a consultant in the John Radcliffe. And our fundamental question has been, what is the impact of ocular disease on human sleep-wake biology? And I won't show you all the data, but I will summarize the findings as they're emerging. The first point, of course, is that, well, visual blindness need not be complete blindness. And of course, many genetic diseases in humans will eliminate most of the rods and the cones, such as age-related macular degeneration, where you lose your cones, Leber's congenital amaurosis, where you lose massive rods and cones, and of course choroideremia, again, massive rod and cone loss. However, although the rods and cones are affected, the photosensitive ganglion cells are intact. And under these conditions, these individuals, they have their photosensitive ganglion cells, and we now need to encourage them to expose their eyes to sufficient daytime light to maintain normal circadian entrainment and sleep-wake timing. In many ophthalmology departments around the world, the ophthalmologist will explain what it's like to go blind, but they'll give absolutely no advice about the regulation of the sleep and circadian systems by this other photoreceptor system. And indeed, it gets even more frightening. They may even encourage individuals to cover up their eyes because they don't look particularly pleasant. And of course, that'll block out the light getting to these photosensitive ganglion cells and disrupt sleep and circadian rhythms. There's even a more extreme case, which is rare but still happens, where the consultant will say, well, you've lost your vision, your eye is therefore no use to you. It's a source of infection, you can't see to look at your eye, why don't we just pop in one of these instead? And of course, unwittingly, we'll remove an eye with photosensitive ganglion cells and the ability to regulate the clock with a prosthetic which looks pleasant but has no biological capacity whatsoever. So completely unwittingly plunging individuals into a world which would be like unremitting jet lag, simply because they were unaware of these new photoreceptors within the eye. The other condition, of course, that one sees in the clinical realm are eye diseases where the inner retina has been lost, including the photosensitive ganglion cells. There's no way for the rods and cones to talk to the brain, and the photosensitive ganglion cells have been lost, and you see this under conditions of acute glaucoma, where the optic nerve and the ganglion cells in advanced stages are completely lost. Under these conditions, these individuals now need to be told about treatments that can help consolidate sleep. So, for example, there's no light treatment that one can provide for these individuals, but one can provide melatonin in the evening, which will act as a signal of the dark, and which has been shown to help align individuals to the light-dark cycle by giving a dark signal via melatonin. Okay. The impact of this could be quite large. The clinical diagnosis of complete blindness must assess the state of both visual and the visual system and the photosensitive ganglion cells. And in most clinical departments of ophthalmology, this is not being done. Eye loss, of course, plunges individuals into a world that lacks both vision and a proper sense of time. And we're not teaching our clinical ophthalmologists of the importance of these photosensitive ganglion cells. Clinical advice in ophthalmology must also incorporate the impact of eye diseases on sleep and circadian timing, completely ignored. And the numbers of patients we're talking about are potentially huge worldwide. There are 39 million completely blind individuals across the world, 285 million with severe visual impairment, and 246 million with some residual vision left. And the question is, we have 500 million people here, and their quality of life and the world in which they inhabit could be greatly enhanced with the knowledge of these new photosensitive ganglion cells. Okay, let me now try and summarize what I've said over the last 50 minutes or so. We talked about the eye and, of course, the capacity of the eye to generate an image of the world using multiple photopigments to detect contrast in terms of the color vision system, and the single photopigment in the rods that allows brightness detection and image detection as a result. We then contrasted this image detection with these photosensitive ganglion cells, and they were discovered because we were interested in trying to understand how the body clock was being regulated by the light-dark cycle. And of course, it's not simply the body clock that is being regulated by these photosensitive ganglion cells, but a whole raft of different other irradiance detection tasks. The photosensitivity is based upon a 480 nanometer, a blue receptor, which is mediated by the photopigment melanopsin. Now, most recently, we've discovered that these two systems, the image forming and the non-image forming, do talk to each other. And in fact, remarkable findings in the mouse that their ultraviolet cones seem to contribute in a very robust manner to the regulation of the light getting to the suprachiasmatic nuclei. And what's going on there, we really don't understand. Luckily, most of the work we found, most of the discoveries in mice have mapped on to humans. Humans have melanopsin-based photosensitive ganglion cells, peaking at 480 nanometers. We did also talk about irradiance levels and the fact that the human clock is relatively insensitive to light, and that raises some important implications in terms of application. Much of architecture, of course, considers the amount of light required for vision, but will completely ignore the higher levels of light required for the regulation of the clock. And then finally, we touched on clinical ophthalmology, and information about these photosensitive ganglion cells is now informing clinical practice, not least to preserve eyes in which these photosensitive ganglion cells still reside, so that people can still regulate their body clock and their sleep-wake cycle. Because without these photoreceptors, you're essentially plunging individuals into a world of timelessness. So the eye, of course, is providing us with our sense of our images of the world and also our sense of time. And with that, I'll thank you all for listening, and I'd like to hand back to Nathan.
Thank you, Russell, for a very, very, very interesting presentation. If anyone is interested, be sure to follow Russell's research. I think they're going to make very interesting headway in that field in particular. Before we go on, I'd like to draw your attention towards this slide. We have the June webinar presented by Serge Dan on evolution on the rotating planet, the importance of the biological clocks. And we also have upcoming webinars in September, October, November, and December. Those can be found on the upcoming webinars page that you all have visited to sign up. Also, I'd like to draw attention to the Luminous Talks that will be happening in the early November this year. Luminous Talks was an event held last year at Parsons New School for Design in New York, and it was a very successful event with very interesting speakers on varying topics on light and health and well-being. So please keep an eye out for those, and please remember to sign up for our next webinar in June. Thank you all very much, and if you have any questions, please feel free to ask those in the Q&A box, and Russell will handle some of those questions and try to answer them as best as he can right now for 10 or 15 minutes. Russell, if you can go ahead and read some of the questions out.
Okay. Right, going from the bottom. We're moving fast from incandescent and fluorescent compact lamps to LEDs, but this type of light is very punctual. Do you think that can prejudice human vision? Well, I think the short answer to that is we need to do quite a bit more research about how artificial light can really enhance both our classical image forming systems and photosensitive ganglion cells. I mean, in terms of the visual system, it's so sensitive to light, often it really doesn't matter particularly which wavelengths you use. You can usually extract some sort of visual image. However, that's not the case with the photosensitive ganglion cells. They do seem to respond maximally to blue light, and those responses we were talking about, shifting the clock, and what we haven't talked about, in fact, is that these photosensitive ganglion cells will also increase levels of alertness, and that is all enormously enhanced by blue light. So the short answer is we need to do a lot more basic biology about the impact of the quality of different light sources in the regulation of not only vision but the body clock as well. Right, I'm just going to scan through and see if there's a few more questions here. So we have a question here relating to our spectral sensitivities. Could it be that there are gene defects in or not gene defects, but gene polymorphisms and such that we might perceive colours slightly differently from each other? And there is some evidence that that is the case. Subtle changes in some of the genes which encode these opsin pigments suggest that some people might have slightly different spectral sensitivities in the green and the blue part of the spectrum. It's only very subtle, however. In terms of the melanopsin system, we have yet to find any individual with a defect in the melanopsin system. They will certainly exist, and we are searching for them in the eye hospital here in Oxford. We have a screen, which I won't bore you with the details, but it's almost certain that we're going to pick up those individuals lacking a functional melanopsin. Could they also have polymorphisms, making them more or less blue sensitive? I don't know. What's quite fascinating is that the melanopsin system of humans, mice, and indeed other groups of vertebrates they're all peaking around about 480 nanometers in that blue part of the spectrum. Now, you find in the visual system, of course, that the rods and cones, or the cones particularly, peak all over the place between different vertebrates. So what is it about 480 nanometers? Why is this blue light so important? And again, I don't have a specific answer, but one can think about the visual ecology of these cells. And if you look to the sky on a perfectly clear, cloudless day, the blue that you'll see is 480 nanometers. And so if you were going to design a system that would be optimally sensitive to environmental brightness, 480 nanometers is exactly where you put those receptors, which is where they're found. So there's a question here relating to eye disease, and so the question relating to macular degeneration. Now, the macula is that part of the eye with the highest concentration of cone photoreceptors. It's where we have our highest resolution within the eye. And what's interesting is that, as we would predict, those individuals may suffer huge visual impairment, but as far as we can tell, their circadian rest activity cycles seem to be perfectly okay. And that's exactly what you'd predict. But it's still important for our ophthalmologists to know this, because even though they can't see very well, they still need to be encouraged to seek out sufficient light to make sure that they're getting enough light to regulate the clock. Now, clearly there will be some circumstances where the eye will show increased sensitivity, and it will be uncomfortable to be exposed to light. And indeed, there are some diseases where additional light may cause more damage. So you can't make a blanket statement. It will depend upon the nature of the eye disease that the advice the ophthalmologist gives. That's why what we're trying to do here in Oxford is to look at a very large number of individuals with eye disease and produce very specific guidelines for ophthalmologists as a result. There's questions about cataracts, which are really very interesting. Now, cataract, of course, is a condition which there's a scattering and a yellowing of the lens. And so what you might predict is that sleep and circadian rhythms would be disrupted in individuals with severe cataracts, because you're filtering out the amount of light getting to the eye, and indeed you're filtering out blue light with the yellowing of the lens. And so we've actually got a lot of unpublished data. Hundreds of individuals have participated in this study, where we've looked at sleep-wake timing before and after cataract operations using a clear lens. Now, the replacement lenses are either clear or they can be yellow and blue blocking. But what I can report is that there's a significant improvement in sleep-wake timing after cataract have been removed and you've used a clear lens implant. We're still looking at the yellow lens data. Again, the prediction might be that the more blue light you filter out with a yellow lens, the more difficult it will be to stimulate those photosensitive ganglion cells and hence regulate the clock. But it depends very much on the type of yellow lens you use, and of course how much environmental light individuals are experiencing. So there's quite a bit of variability, but we'll have a clearer answer to that in the next few months. So there's a question here about the lady who we did the testing with. You know, can you see the light or not? Could she just be guessing right? It's a very important question, and that's why the studies were pretty rigorously undertaken. If you were to guess by chance, you would have to. I think you'd have to. Well, you'd be right 10 times out of 20. To get statistical significance, you'd have to get 15 times out of 20 right. And this particular lady would get it 20 out of 20 times. She always knew, without realizing how, that when the lights were on and when the lights were off. So you have to use, significant statistical power to elucidate her ability to detect the lights were on or off versus chance. Okay, I'm just looking through. So the question is, do you think information about this third photoreceptor will actually get out there? What we're trying to do is produce a robust set of guidelines, and we are now collaborating with ophthalmology departments around the world. So I think it's slowly getting out there. I certainly remember giving a presentation only 5 years ago to a group of European ophthalmologists, most of whom still had no idea about these new photoreceptors. In fact, one individual came up to me, one consultant came up to me afterwards and said, "My goodness, I have scheduled on Monday a bilateral removal of the eyes of this particular patient, and I have no idea, and they are visually blind, but I have no idea whether those photosensitive ganglion cells are working or not, and I've never thought about the impact of the eye on the body clock at all." And it's worth pointing out that not having properly adjusted circadian rhythms is more than just the inconvenience of going to sleep and getting up at the wrong time of day as your clock drifts through time. Disrupted circadian rhythms, as in shift workers, have been associated with a range of additional pathologies. So, for example, cardiovascular disease, immune suppression, metabolic abnormalities are much more prevalent in individuals with a disrupted body clock, such as shift workers, or indeed people with jet lag. So it's more, getting your body clock right is more than just stabilizing your sleep rate timing. It's overall health as well. I'm afraid it looks as though we've run out of time. I very much appreciate your attention and all of your questions. And please don't hesitate to get in contact with me if there are questions which I haven't got to and you'd like answered. Once again, thank you very much. Goodbye.
Thank you, ladies and gentlemen. Thank you again, Professor. That was a very, very interesting set of questions that were answered, and I hope everybody got the answers they were looking for. As you mentioned, please don't hesitate to get in contact with him if you still have questions that have been unanswered. And remember to look forward to our June webinar by Professor Serge Dan. And have a great day.